My name is Dale Overton, and I'm a passionate soil scientist and entrepreneur. And today we're going to talk about soil ecology and why it's important for farmers and gardeners to consider soil ecology when they're planting their gardens and their crops. Soil ecology just refers to the interactions of all of the organisms that live in the soil with one another in their physical environment. So, in standard conventional agriculture, there's really a discounting of what microbes actually do in a natural system. You feed your chemicals to your plants, and those plants will actively take those chemicals up, and it discounts the role that microbes play. In a natural system, if you take humans out of the equation, for example, walking into a forest, there's no chemicals there, right? It's an entire ecosystem of microbes and plants interacting with each other that releases nutrients, suppresses disease, and creates that biomass. In agriculture, we've really forgotten about how microbes can actually impact healthy crops. And I think it's important for farmers to really understand that building organic matter back into our soils and building soil structure will really help us to reduce the amount of chemicals that we need to apply to our crops to get a healthy crop, but also to take care of that land so that we can actually continuously crop that land. We know more about the moon than we know about the soil from a microbial standpoint, but we really know that there's a huge complement of microbes that interact with plants that plants communicate with. So when we talk about functional microbes, we're talking about things like mineralizing nitrogen or fixing nitrogen from the atmosphere. Many farmers will input a huge amount of nitrogen into their system to get that rapid growth. But in a natural system, there's actually bacteria that will mineralize it from the organic matter and then also mineralize it from the atmosphere. Most of the gas in the atmosphere is actually nitrogen. So that's an example of a function. Plants also require phosphorus. So in a natural setting, when a plant requires phosphorus, it sends out signals through sugars and other exudates into the soil that will actually trigger specific organisms that release phosphorus. And they basically release organic acids that unlock phosphorus from the soil colloids or from clay. And that's something that, again, is an extremely important function because phosphorus is typically the main limiting element in any natural system. So a huge amount of phosphorus is typically added in agriculture, although there's a huge amount of phosphorus already in the soil. So we're adding phosphorus when we already have phosphorus. We're simply missing that microbial bridge, that function to unlock the soil phosphorus. And when we start to understand how that works and how to use those microbes to get that, we can actually reduce our phosphorus inputs and our nitrogen inputs significantly. There's an unfathomable number of microbes in the soil. So you'll often hear that when you talk to an agronomist, there's, billions of microbes in the soil. It's really about who's doing what. And yeah, there's bacteria, there's actinomycetes, there's archaea, there's fungi, there's a whole, complement of organisms. And within each one of those groups, there's potentially tens of thousands of species. And really, they're all doing different things. It's a complex network. It would be very similar to a soil food web. Well, it is a soil food web, frankly. So you've got fungi that will converse with bacteria that, they exchange resources quite often. In a lot of ways, it's kind of like warfare. Everybody's trying to fight to get resources, and ultimately it's going to be the plant community that's going to select for what microbes are really there because they require certain things that certain microbes will provide. And then you have the auxiliary community. So you've got your organisms that will degrade organic matter. So you've got fungi, for example, that will release organic acids and break down carbon compounds. So if you've got a lot of corn trash, or if you've got a lot of wheat stubble or hemp stubble, you can actually use fungi to break down that organic matter without sucking up a lot of the nitrogen out of your system. So that's just one example of a type of microbe. There's also fungi that are called mycorrhizal fungi that form symbiotic relationships with plants. So, upwards of 90% of all plants actually form symbiotic relationships with mycorrhizal fungi. And they basically just form an extension of the plant's root zone. and provide functions that the plant can't necessarily provide for itself. And mainly, that's going to be phosphorus mobilization and uptake. It's an ebb and flow type of system. So if you've got an imbalance in the system is imbalanced. So in many cases, conventional agriculture and its core has disrupted the system. So when you take a look around, every single conventional field in North America is a case for reclamation. We've completely discounted what the microbes are doing. A lot of those functions have since disappeared. So if you plant a crop, that crop can no longer function on its own because the functional microbes that are responsible for creating that healthy system and that healthy plant are gone, right? They just don't come back. It's not something that, I mean, you can use inoculants and whatnot to bring some of those functions back, but if you're looking at overall soil health, there's a whole bunch of things that would have to be done in order to really bring that community back. So yeah, it's not just about one or two species, it's an actual functional community of microbes that are required for not only plant growth and development, but also just in general soil health. So water retention, nutrient cycling, nutrient holding capacity, all of these things are really controlled by microbes and the way that soil is structured from the activity of microbes. Pillage basically ravages a huge number of what I'll call fungi and mycorrhizae. They form, they have a mycelium, so they have roots like plants that are very, very fine that, in some cases will go for kilometers. And when you till your soil, you're basically breaking those up into fragments and destroying that network. So you really will lose that whole fungal functional aspect when you till. Other things, glyphosate. Overuse of glyphosate and other herbicides really impacts, in a lot of cases, more bacteria than fungi. So glyphosate impacts the shikamat pathway, which is a vital pathway for bacteria. And when you use glyphosate and it actively gets on a lot of these bacteria, they'll die, right? They can't function anymore. So you see a real phase shift in typically your healthy bacteria, your good bacteria. I mean, some bacteria can withstand it, but most bacteria will be negatively impacted, and it just so happens that the important bacteria are the ones that get the most impacted. And when I say important, I'm talking about important functionally for plants and plant growth and development. Diverse cover cropping is probably the easiest way that a farmer can really start to rehabilitate their soil. And in that, I'm talking about between crops. So, for example, when you pull off your wheat in late August, if you were to plant a cover crop, a 7 or 8 species cover crop that could grow in the shoulder season, you're always keeping that soil covered. So that means that that soil is always functioning. You're building stable soil aggregates. You're increasing its ability to cycle nutrients. You're increasing the ability for that soil to actually carry those communities forward for the most part. So that would be the first step, minimizing tillage. I'm not saying no tillage, but minimizing it. Obviously, in certain circumstances, it's a requirement. But, don't deep rip your soils, and again, cover crop. And diverse crop rotations, I think, are another thing. So you're using like at least a 3 to 4 crop rotation. The idea is you want to get cover crops that actually have diverse root architecture. So you've got some that have a taproot, you've got some that have a really fibrous root system, and some that have, affinity for mycorrhizae. Bringing cover crops into the equation, especially from a diversity perspective, really helps to increase the ability of that soil to function in the long term. And it's a perpetual thing. It's not just like you cover crop one year, it's a continuous thing. And I think that, the input cost for cover crops is definitely offset by the soil's ability to hold water and cycle nutrients. And what that will do is minimize the amount of inputs that farmers need to use. And I'm going to say while maintaining current yields, right? Everybody always wants more and more and more, but there's really only so much that your soil can do. And I think that if you can maintain your current yields while reducing your inputs, you will create that sustainable farming system that, this whole regenerative agriculture scene is trying to get at. Well, I think that there's a lot of great inoculants on the market. Single species, multi-species. In my mind, very similar to what I was just saying about cover crops, I think it's the diversity that's really important. And, you can put 1 or 2 species of microbe down that have a couple of functions, but you're not going to get that whole, again, it's an ecosystem. So ecosystems aren't comprised of 1 or 2 species. They might be comprised of a keystone species, but you have a whole, subset of other functional groups that are really important. So, things like Bacillus subtilis or Pseudomonas petita or Pseudomonas fluorescens or, some of the mycorrhizae from the glomales family, which are arbuscular mycorrhizae. You can get all of these as independent inoculants. But I think the real goal here is to try to create a very low-cost method to actually get all of those microbes and all of that functional diversity into one place. Frankly, when you're dealing with single species inoculants, they might work in Florida really well, but they don't work in Manitoba really well. So you don't get that ecological dexterity that you would get, with more natural type of products from a broth culture or what have you. In conventional agriculture systems, you've killed off a lot of your really beneficial microbes. So the first organism to kind of come into any stressed system is going to be a pathogen, right? That's because the natural immune system has kind of been compromised. So one of the things that a diverse biology does is it really reduces the amount of pathogens that can actually inhabit a soil. And again, that's through specific antagonism, which is just competition for resources and space. But the other is actually the production of antibiotics. And, in my mind, that's one of the coolest things that, soil microbes really do is they create these antibiotics that are actually specific to pathogens and other organisms. Again, they're trying to fend for themselves in an environment that's extremely hostile. So there's a whole plethora of organisms that produce antibiotics. Penicillium is one of them, right? Penicillin, it's probably the most famous antibiotic that's ever been made, was isolated from an organism that's prevalent in the soil. You've got bacteria like Streptomyces, which is a type of bacteria that lives in the soil. They form roots kind of like fungi, but some of the most potent antibiotics in the world have been found in that genus, and a lot of them are actually associated with earthworms and healthy plant systems. So it's really cool. Once you get that diversity, you start to see a huge reduction in pathogens. Not like they don't exist, but maybe not enough to impact your crop as much as you might think. There's a lot of pathogens that I'll call that are soil-borne. There's a lot of pathogens that are blown in. So, for example, like Phytophthora in potatoes is typically blown in, or it comes from a contaminated plant. So foliarly, you want to try and create a system where, like, a lot of microbes will actually colonize the leaf surface. It's called the phyllosphere. So if you actually spray a microbial concoction onto your plants prior to, a really intensive disease scenario, those organisms will actually help to fend off those plants from any disease by colonizing the leaf surface. And in a lot of ways, through the same mechanisms that we just spoke about, through the production of antibiotics, through competition for resources, they'll actually suppress pathogens. And in that case, really reduce the amount of fungicides that we actually put onto our crops. The nice thing about biology is you can combine it with foliar nutrition. So you can really address a lot of the nutritional requirements of the plant while inoculating it with microbes, very similar to a fungicide. You can also mix a fungicide with nutrients, or a herbicide for that example, with foliar-based fertilizers. You don't really ever want to mix herbicides with microbes because their modes of action are a little bit different. So, in some cases, it takes a bit of ingenuity in order to use microbes in a conventional farming system. And I think that's where this whole regen ag thing is kind of coming in, because it's not like organic agriculture where you're confined to using only natural products. I think in the regenerative approach, you can still use pre-emergent herbicides. You can still use a fungicide if you have to. You just really have to start practicing more regenerative soil techniques. So cover cropping and, reduction in tillage and those kinds of things. A big part of that immune system is its relationship with the plants, with the microbes that occur in the rhizosphere. So for example, having that really healthy rhizosphere is an extension of the plant's immune system, because it keeps pathogens away, it keeps that soil functioning so that plant can maintain really good health. If you have a really healthy rhizosphere, really healthy microbial community in your soil associated with your plant roots, you'll actually improve the ability of that plant to create sugar through nutrient use efficiency and uptake. And increasing that sugar, those Brix levels in your plant, will help to reduce insect pressure, because insects don't have a pancreas, so they can't process sugar very well. Plants that are fed more chemically, they're taking up more water, they have less ability to create those really high Brix levels. So you see really significant potential for insect attack and then secondary infection from different pathogens. When you have a really healthy functioning rhizosphere and also on the plant surface, those insects just can't necessarily spread the disease or impact that plant nearly as much. They'll, in a lot of cases, they'll go to your neighbor's field, or they'll impact other places before they'll come and impact you. There's not a huge amount of knowledge out there on how the phyllosphere microbiome will impact photosynthesis, although it does help to, impact some transfer through the leaf surface, through the stomata, but mainly that's going to happen through the soil. It'll be soil-borne microbes will impact photosynthesis, I think, more than organisms on the phyllosphere. You can probably see minor corrections during the growth season, or during the growing season. But again, I think that foliar feeding is really kind of like a spoon feeding approach. And you're going to see most of your nutrient transfer coming in through the roots. You can till a little bit, but if you deep till and you ruin those fungal relationships, you ruin the soil's ability to recycle organic matter. And I think that that's probably the biggest problem in modern agriculture, is the way that we actually manage our crop residues. Burning crop stubble because it's a problem is one of the reasons we're in this mess in the first place, because that's basically your soil's organic matter. You're taking organic matter out of the ground to create biomass. You pull off your grain, but you leave your stubble behind. Unless you incorporate that stubble properly, you're really going to run into issues with depleted organic matter. Tilling is another reason why that happens. So what ends up happening is, through tillage, through fungicides, you actually kill off a huge component of the biology that's important for reintegrating it back into the organic matter, or recycling it. So for example, in most conventional fields that lack fungi, bacteria will actually consume a lot of that organic matter, but in doing so, they suck out nitrogen from the system. So what ends up happening is you have to account, let's say 20% of your nitrogen budget for the whole year is accounting for the amount of energy and resources that those bacteria require to break down that carbon. Now, if you actually have a fungal community present, those fungi will actually break down that organic matter in the absence of nitrogen. So you're not sucking up a huge amount of your nitrogen from the system. They're releasing organic acids to break it down, and then reintegrate it back into that soil organic matter. And what that's going to do is create a healthy soil aggregate, and continuously create healthier soil aggregates and recycle that organic matter. So what happens is you'll see a reduction in the loss of organic matter. Well, you'll need to use a fungal inoculant, frankly. I mean, the only way to really get fungi back into the system is to reintroduce them through inoculants in a variety of forms. Fungal composts are a great way to get them. But when you do spray them, yeah, they do carry over. Absolutely. As long as they're on that residue, they'll dormant over winter. And then as soon as spring temperatures rise above zero, they start to work. And again, it's the nice thing about that is those fungi will also help to suppress other diseases. A lot of diseases actually live in that residue, right? They're carried over through crop residue. So the better you manage your crop residue with beneficial fungi, the less you're going to carry over disease into the following cropping year. When you integrate that with a diverse cropping system and cover cropping, disease will become a thing of the past for most farmers. Now, it's a different way of managing the system, right? It's a different way of thinking about it. But, if you look at guys like Jay Fuhrer at Minoculants Farms in North Dakota, they're completely regenerative. They haven't used a fungicide in, 10 years? And they're getting great yields, and they have great soil structure, and they use very little nutrients. If the ground's frozen, then yeah, you probably wait till the spring, but if you could get it on after you chop down where there's a will, there's a way, as far as I'm concerned. So you can always, put it down in your fall working. When you work your fields in the fall, that's when you would put it down.
Does fall rain help incorporate biology into the soil?
Yeah.
So
it will, as long
in a misty day.
Yeah, absolutely. As long as you've got good soil structure, right? Because if you got a lot of moisture, it's going to sit on the surface, and then you're not going to get a lot done. But if you got good drainage and you can get it down, yeah, it's rain will help as a carrier, right? So it'll help just spreading it and making sure that you've just got homogeneous distribution of whatever it is that you're putting on. Sunlight is an enemy, right? Sunlight really desiccates and impacts soil biology. So yeah, I always recommend a light harrow or a light till event post inoculation of a residue product, simply because these organisms live in the soil. So you want to get a little bit of soil contact. You want to kind of bury that trash a little bit if you can. When you think about the forest, you go in there and there's like a bit of a duff layer. So the surface of the duff layer where the fresh stuff has fallen, there's not a lot going on in there. There's a little bit, but as soon as you go down just a little bit further, you can pull it up and you'll see all sorts of stuff happening. There's fungi, there's all sorts of stuff. But again, it's kind of out of the sunlight and it's a little bit further down. So I would highly recommend a light harrow just to get a little bit of soil incorporation. When you have a really healthy biological system, the nutrients that you do put down are going to stay there. So a lot of guys will say, those bacteria or those fungi are robbing your plant of the nutrients. And I would say that that's actually incorrect. What they're doing is they're holding those nutrients in the soil and allowing that plant to have long-term access to those nutrients. So they prevent volatilizing, they prevent leaching. As far as compatibility goes with like a tank mix or putting it in with a foliar, if you got salt-based fertilizers, it's always good to stabilize those with humic or fulvic acids to kind of chelate those nutrients, and then you can actually create a much more hospitable environment for a lot of microbes. I'm not saying that it's perfect, but you can definitely tank mix microbes in with chelated nutrients. But humic acid, fulvic acid, I would stay away from the EDTAs and some of the other chelating agents that are typically used in chemistry. They're food sources for microbes because they chelate the nutrients. So, and they're organically based, so they actually provide a really good food source for microbes. Humic acid, there's 20 different organic acids that form the humic acid family. Some of them have high molecular weight, some of them have low molecular weight, so they have different functions. It's not just humic acid. So there's a bit of a misconception about that. But ultimately, humic acids are naturally occurring in the environment through decomposition, and they can be used as an input to improve soil function. Now, when we talk about soil function, if you're putting them down in the absence of having a good microbial community, they're not doing a whole heck of a lot outside of chelating nutrients and supporting that biological community that's already there. If you have a really healthy biological community, or you're inoculating it with that community, you just get much more benefit for, get much more bang for your buck, so to speak, simply because you're putting that microbial community down with the humic acids. So they work in conjunction together in the soil. So chelating basically just means binding up. So it's like a lock and key model, where they, certain nutrients will have an affinity. So, if you got really high salt-based fertilizers, like if you're putting urea in, when you mix urea with humic acid, you'll bind up that nitrogen. So nitrogen is typically volatile. It'll leach out of the soil very easily, or it'll gas off very easily. When you chelate it, you basically take, another chemical and bind the nitrogen to it. So it immobilizes that nitrogen, but through a fairly weak bond. So that nitrogen, or whatever the element is, can actually be accessed by plants and microbes fairly easily without a huge amount of energy being expended. I would say lighter soils would definitely benefit a little bit more, simply because, sand doesn't, like clays will, just the natural affinity of a clay to bind cations is huge. So, just the overall cation exchange capacity of a clay soil versus a sandy soil is, is huge. So humic acids would definitely have a higher benefit in a sandier soil. Any kind of microbes you'll use will typically have a higher benefit in a sandy soil, simply because they're fairly low in organic matter. They leach fairly easily, so there's just not a huge habitat there. But using humic acid in biology, you can kind of transform those soils into a much more sustainable type of system, right? By integrating the organic matter, those kinds of things, you can actually transform sandier soils into more functional soils. There is more going on than what we know, and ultimately, I think education is the biggest tool in helping farmers to understand how they can improve their net returns using biology. Because all your money doesn't have to go to chemical companies or companies like ours. You can use biology on the farm for a very low cost and see significant benefits and returns.